Today topic of discussion is Kreb cycle. Kreb cycle also called as TCA cycle or citric acid cycle. Name after scientist, it have that name Kreb cycle. It have 3 carboxylic acid component. Then the first component is citric acid. So it have 3 different type of name. Pyruvic acid. This pyruvic acid we get from glycolysis. Glycolysis, glucose is converted into 2 component of pyruvic acid. That pyruvic acid enter to citric acid cycle. So pyruvic acid is converted into acetyl-CoA. Here couple of changes takes place. One carbon given up in the form of carbon dioxide. Reduction reaction will occur. NAD plus is converted into NADH2. Okay, this reaction is not considered as a part of cycle. This one is a oxidative decarboxylation reaction. So pyruvic acid 3 carbon component is converted into acetyl-CoA. Here one carbon given up in the form of carbon dioxide, so we get 2 component. Here coenzyme added, so acetyl-CoA. This acetyl-CoA accepted oxaloacetic acid with the help of citrate synthase enzyme. It will converted into citric acid. Acetyl-CoA 2 carbon component, oxaloacetic acid 4 carbon component. So it will form 6 carbon component citric acid. It is a first product, so this cycle have name citric acid cycle. For our convenience we are giving name. This one is the first one. This one is an oxidative decarboxylation process. So this NADH2 not considered for ATP calculation. This one is the separate. We will counting here only. Then citric acid is converted into isocitrate with the help of enzyme isomerase enzyme. Then isocitrate is converted into alpha ketoglutarate. Here also one carbon component given up in the form of carbon dioxide. Reduction reaction will occur. NAD plus is converted into NADH2. Then alpha ketoglutarate is converted into succinyl-CoA. Here coenzyme A added, one carbon given up in the form of carbon dioxide. Here again that reduction reaction will occur. NAD plus is converted into NADH2. So 2 NADH2 molecules we got. Then this succinyl-CoA, here also carbon dioxide produced, no, so we have only 4 carbon component. Citric acid 6 carbon, alpha ketoglutarate 5 carbon, here 4 carbon, 2 carbon given up in the form of carbon dioxide. Then this succinyl-CoA is converted into succinic acid. In this reaction some energy released. This energy is given up, used to convert GDP to GTP. Then ultimately give this GTP ultimately given one ATP molecules. Then this succinic acid, here coenzyme A given up. Succinic acid is converted into fumarate. Here also reduction reaction will occur, but the substrate here not NADH2. Here FAD is converted into FADH2. What is the difference between NADH2 FADH2 means NADH2 entered to electron transport chain means it produced 3 ATP molecules. FAD enter electron transport chain means it produced 2 ATP molecules. Okay, then the fumarate is converted into malate. Then malate is converted into oxaloacetic acid. Here also reduction reaction will occur. NAD converted into NADH2. Then this oxaloacetate accepted the acetyl-CoA, then it forms citric acid. It will continue that Krebs cycle. So summary, how many NADH2 to give here? 1, 2, 3. So totally 3 NADH2, then 1 FADH2, 1 ATP molecules. Here different type of enzyme involved. First one is citrate synthase, second one is isomerase, third one is coenzyme A. Okay, the coenzyme A. These are the enzyme involved in this reaction. So how we will calculate ATP? Krebs cycle we already known. Glycolysis, one glucose molecule will produce two pyruvic acid. So one pyruvic acid enter to glycolysis means it produced 3 NADH2. 3 into 3 NADH2 entered to electron transport chain. It produced 9 ATP molecules. 1 FADH2 means multiply into 2 ADP molecules, 1 ATP, 1 ATP. So totally we get 12. We have 2 pyruvic acid, so we will multiply into 2. So totally we will get 24 ATP molecules. Krebs cycle we will get 24 ATP molecules. Now we will see that notes format. Pyruvic acid change into acetyl-CoA, acetyl-CoA into citric acid, acetyl-CoA accepted by oxaloacetate to form citric acid, then isocitric acid, then alpha-ketoglutarate, then succinyl-CoA, then succinic acid, then fumarate, malate, then change to oxaloacetic acid. This cycle continue. Finally we got 24 ATP molecules. Thanks for watching.